Scientists have just made one of the most important medical breakthroughs of our lifetime. Look, I know that sounds like hyperbole, but I promise it's not an exaggeration. Just stay with me for a second. Meet KJ Muldoon. He was born with a rare and life-threatening genetic disorder. But in February of 2025, at just 7 months old, KJ received a treatment that was truly unprecedented. The world's first ever personalized gene therapy. Think about that for a second. Up until now, medicine has used a one-size-fits-all type of approach. You get a diagnosis and then a doctor prescribes from a limited menu of treatment options that are designed to treat a general population. Chemotherapy for cancer, insulin for diabetes, broad-spectrum antibiotics for bacterial infections. Up until now, these treatments often manage the disease well, but they don't work the same for everyone. The idea of personalized medicine, a treatment crafted specifically for your genetic code to fix your individual biological error, has been somewhat of a scientific mirage. Tantalizing vision so close you can almost touch it, but up until now has only stayed within the confines of scientific and medical journals. Until now. This is the incredible story of baby KJ, the groundbreaking science that saved him, and the dawn of a new era in medicine. What's up, nerds? In this video, I'll mostly focus on the science behind this groundbreaking discovery and the impact that it'll have on personalized medicine and medicine as a whole. But we can't talk about personalized medicine without talking about the person. KJ Muldoon entered the world in August of 2024, a day that for his parents began with joy and excitement. However, within his first weeks of life, their world shifted as doctors discovered a severe problem. KJ had a life-threatening genetic disorder that affected his liver's ability to process protein. A disease that, if untreated, can cause severe and irreversible brain damage, and tragically, can be fatal. To truly understand the gravity of this diagnosis, we need to take a step back to understand what genetic disorders are, and in fact, what genes and even DNA is. If you're on this channel, you probably know what DNA is, but in case you need a reminder, deoxyribonucleic acid, or DNA, makes our cells' genetic code. Think of it as a detailed instruction manual for building and running practically all of the cells in our body. Specific segments of DNA are called genes. Each gene contains the instructions for making messenger RNA, which quickly gets translated into a particular protein. The central dogma of molecular biology, baby. Proteins are the workhorses of our cells, performing countless essential functions from building tissues to carrying nutrients. A genetic disorder occurs when there's a typo or an error in this DNA instruction manual that leads to a faulty or missing protein. Faulty or missing protein means faulty or inactive cell activity. KJ's specific genetic disorder led to a deficiency in a protein called carbamoyl phosphate synthetase 1, or CPS1. To put it simply, his body lacked this crucial enzyme, which is a vital component of the urea cycle in the liver. The liver is a very important character throughout the story, so don't forget it. The urea cycle is one of our body's natural detoxification pathways, responsible for converting highly toxic ammonia, a byproduct of protein metabolism, into harmless urea, which can then be safely excreted. For individuals with severe CPS1 deficiency like KJ, if there's not enough of this CPS1 to process this ammonia, it builds up to dangerous levels in the blood, and if untreated, can cause severe and irreversible brain damage, seizures, coma, and tragically, can be fatal. Studies have shown that severe neonatal onset CPS1 deficiency carries a high mortality rate, anywhere from 20 to 50%. Without a functioning urea cycle, the prognosis is often grim, with many infants not surviving the first few weeks or months of life without aggressive intervention. Those that do survive often face significant developmental delays and intellectual disability. What options do parents have for their children who have this CPS1 deficiency? And to be honest, there aren't many. Historically, the slim options include reducing the amount of protein in their diet to mitigate how much ammonia is built up, or to undergo a liver transplant. A liver transplant is the primary intervention because it has the longest positive effect. The rationale is if your liver can't make enough CPS1, simply get a liver that can. And while a liver transplant can be life-saving, it's a major surgery with significant risks, especially for an infant. Younger children face higher risks of surgical complications. For example, infections are reported in over 50% of pediatric liver transplant recipients. There's also a lifelong need for powerful immunosuppressant drugs, which come with their own set of side effects, including increased susceptibility to infections and kidney dysfunction. For a condition as rapidly progressing and dangerous as a CPS1 deficiency, time is of the essence. And with the current treatment options being less than optimal, KJ and his family needed to turn to something better. They turned to something called gene editing. This revolutionary technology goes straight to the source by correcting the typo that's in the DNA of KJ's liver cells. No transplants, no low protein diets, just a single edit that adds the genes to make a fully functioning CPS1 enzyme. This brings us to the next chapter in KJ's story. We're we're going to focus a lot on the technical aspects of this gene editing technology. It's a safe bet this will probably be the future of genomic medicine, so it's good that we all know about it now. Let's start with the specific gene editing technology that you've probably heard a lot of by now. Clustered, regularly interspaced, short palindromic repeats, or CRISPR. It acts like scissors to cut and change certain parts of DNA within a cell. CRISPR consists of two main components: a base editor, which is an enzyme that does the actual snipping, and a guide RNA, which acts like the hands to, well, guide the base editor scissors to the appropriate place to snip. And so in KJ's case, scientists wanted to design a CRISPR system that would specifically edit the tiny genetic typo in KJ's liver cells, cut the bad gene out, and replace it with the correct gene that produces a normal amount of CPS1 enzyme. CRISPR has been an undeniable success in the world of genetic engineering over the last few decades. It's way faster, cheaper, and accurater than old methods of gene editing. Several CRISPR-based technologies have been FDA approved in recent years for treating diseases like sickle cell disease, beta thalassemia, and hereditary angioedema. We know that CRISPR can work really well in humans, and it can be used to help KJ, but past treatments were general, a one-size-fits-all treatment for all patients with that disease. Also, CRISPR isn't perfect. Sometimes you can snip off more DNA than expected, leading to many unwanted insertions and deletions. It can cause more problems than it solves. But KJ didn't need a large cut. When scientists extracted DNA from his liver cells and analyzed his genetic code, they saw that he just needed a slight change. A change in a single nucleotide, or a single letter in his entire code. And this is where the medicine really starts to get personal. Scientists needed to have the utmost precision when trying to fix KJ's single letter mutation. Rather than acting like scissors, this personalized treatment was like using a very fine eraser that could avoid any of the downstream issues caused by larger cuts and double-strand breaks. So scientists extracted KJ cells and tested this in a dish and saw that it worked amazingly. After some iterations, the edits happened exactly like they planned them to. But using this little eraser on KJ cells in a dish is not the same as using it in a person. But how is this CRISPR system actually delivered to his liver? Scientists made a lipid nanoparticle, or LNP. They look something like this: a combination of lipids that encapsulate both the base editor mRNA and the personalized guide RNA. We've talked about LNPs ad nauseum on this channel, so I've linked a video down below that talks about them in great detail. Don't forget to give that a like, come back here, give this video a like, and subscribe. Thank you so much. But in summary, LNPs serve to protect the CRISPR components from being degraded by various parts of the body that detect foreign nucleic acids. And they deliver these CRISPR components straight to the liver. LNPs are just like an Uber, safely delivering precious therapeutic cargo to the right address. Hell, make it an Uber comfort. Extra legroom, chilled water bottles in the back, no threat of destructive endonucleases during circulation, the works. And the best thing about LNPs is that they love to go to the liver. Binding of various liver-targeting proteins and immune cells to the surface of LNPs means that over 90% of administered LNPs get brought to the liver within an hour. And in many other pharmaceutical applications, this is a problem because it stops them from working properly. But for someone like KJ who needs treatment in the liver, it was an absolute no-brainer for these scientists to use LNPs to maximize the delivery of KJ's custom CRISPR. A custom base editor and guide RNAs to treat an unbelievably rare genetic disorder, all packaged in a nanoparticulate Uber driver. Sounds like a fever dream, but it's actually just science. Really damn cool science. But let's not forget that science has to be more than just cool. It needs to actually work. Now it's time to finally answer the question we've all been waiting for. As of April 2025, KJ has received 3 monthly doses of his custom CRISPR LNP with astounding results. His protein intake had increased, his dose of nitrogen scavenging medication had decreased. He's even been able to fight off common viruses without complication. He's starting to be like a normal kid again. All that would have been impossible just 3 months prior, and all without any side effects. Of course, KJ will need to be monitored continually to ensure there are no side effects in the future. He'll probably need to be monitored for the rest of his life, and will probably need more doses of this CRISPR LNP to make sure that his liver continually and stably produces enough CPS1. But regardless, it is undeniable how big of a feat in modern science and medicine this is. A child, barely 6 months old, gets a treatment that's tailored for him. And I just want to take a second here to look at how efficient this whole process was. In just over 3 months, Dr. Arens Nicholas and Dr. Musunuru, the 2 lead scientists on KJ's treatment, analyzed KJ's DNA, made a personalized treatment, and did preliminary in vitro and in vivo studies. This pace is absolutely mind-blowing. Typically, this stuff takes years and years of development, but this is a great example of how quickly great science can be done with strong collaborations and with good funding. You know who you are, people who want to defund biomedical research. Now, as we wrap up here, I want to address some of the pessimists out there who are probably thinking, well, if we have the technology to do this, imagine what would happen if it gets in the wrong hands. And I see your point, like I really do. CRISPR could be used for evil, and it could be used to recreate the 1997 sci-fi classic Gattaca. But it's up to everyone, not just scientists, to make sure that this technology is used for the right reasons. Luckily, one of the inventors of CRISPR, Jennifer Doudna, has stressed the importance of strict guidelines to ensure that it is used properly. She recommends that we focus on using CRISPR-based medicines only in somatic cells and not sex cells. You see, KJ's custom CRISPR treatment won't automatically pass his gene-edited liver to his offspring, because the edits were made directly to his liver. In order for it to be passed on, this edit would need to be done in his sex cells. But this current limitation is probably necessary, because it also means that people can't harness this power to simply edit your babies to have a certain eye color before they're born. Now, I'd like to think most people are reasonable and they won't use CRISPR for evil reasons. And even if you're using them for the right reasons, to make sure that you go through the right safety and ethics protocol. Many are probably familiar of a certain scientist named He-Jen Kui, who tried to treat HIV-susceptible children with CRISPR technology. There are many other videos described in this case, so I'll leave it to them, but in general, if you're gonna make a treatment like this, just make sure you pass ethics first and that all patients are willingly consenting. But let's go back and focus on the personal aspect of this technology. I don't have kids, but if you ask any parent, they'll say the first and only thing they wish for is a happy and healthy baby. I can't even imagine what KJ's parents and all the other parents who receive similar news, hearing the news that your newborn baby has a life-threatening genetic disorder, but now less than a year later, they can see their happy and healthy baby and know that he is a beacon of hope for all children who have a very rare genetic disorder. I usually like to keep it pretty light on this channel, but I can't deny that when I first heard about this story, it brought a tear to my eye. And if you want a lot of tears to come to yours, I'd highly recommend watching this video. They interview the Muldoons and the doctors who made KJ's treatment. It has the gravitas and emotion that a scenario like this deserves. A link to the video is in the description. When you're done watching that, feel free to come back here and let me know how much you cried. That's all for me today. Thank you so much for your time. And if you like this video, feel free to give it a like, but more importantly, share it with someone who you think needs to hear a little bit more about optimistic science today. If there's other groundbreaking or optimistic science that you'd like me to cover, feel free to leave it down below. I'd like to leave the last words of this video to Dr. Ahrens Nicholas and Dr. Masanuru, the lead scientists on this study, where they give their views on the future of personalized medicine.
We hope this is the first of many. I have so many patients with huge unmet need, right? So many patients where there's not a good option available for them. But I really hope that 10 years from now, targeted personalized therapies are available for most of my patients. That would be the dream.
Oh, for me, it's more than hope. It will happen. No, it will. I mean, now that we have shown the way, I think this is the start of something.